I is anterior rami. It's been a long time here. In this video today, we want to look at development of central nervous system. You get? And when we talk about central nervous system, we all know that it involves the development of the brain and the spinal cord. Development of the central nervous system occur through a process known as neurulation. Yeah, and it occur around the second and third week of development. Though the entire complete formation of the neural tube, you get, the tube that is going to give rise to the central nervous system occur around the third week of development. So we want to look at development of neural tube, and this is the development of central nervous system 1. So in this video, I'm going to emphasize more on what neural tube is all about. Yeah, so let's continue. Now, if you don't forget, in trilaminar embryonic disc, you get, there's something we call the ectoderm, which is the outer layer. We also have the mesoderm, which is the middle layer, and we also have the endoderm, which is the what? Inner layer. Yeah, so this is the ectoderm. Yes, let's call this as the ectoderm. Then, why this middle part is what we call the mesoderm, and why this, let's use this as the ectoderm. Yes. Then, this part here is what we call the endoderm. So, this is the ectoderm, this is the endoderm. Alright, let's continue now. So, in between the ectoderm and the endoderm, there's something we call the mesoderm. You get it now? So, when we talk about the mesoderm, there's a structure. There's a structure lying in between the mesoderm, and that is what we call the what? The notochord. So, this is what we call the what? The notochord. Yes. So, the notochord is just like a structure that is extending between the ectoderm and the endoderm. So at either side of the notochord is what we call mesoderm, and these are the mesoderm that we are. These are the mesoderm. Yes, they are bilaterally located at either side of the what? Notochord. So this mesoderm that lies directly to the notochord is what we call the paraxial mesoderm. You get? We call this what we call what? We call it paraxial mesoderm. This is what we call paraxial mesoderm. And this mesoderm that is lying next to the paraxial mesoderm is what we call the what? Intermediate mesoderm. So this is what we call the intermediate mesoderm. Yes, intermediate mesoderm. And there is a particular structure here also. It is also mesodermal in origin. This is what we call the what? Lateral plate mesoderm. But there is a structure that divides the lateral plate mesoderm into two. We call it the intra-embryonic coelom. It is the one that divides the lateral plate mesoderm into two. It divides it into a somatic layer and a splanchnic layer. So this part of the lateral plate mesoderm is what we call somatic lateral plate mesoderm. And this part is what we call the what? Splanchnic lateral plate mesoderm. This structure will help us in the development of the central nervous system. This is what we call a trilaminar embryonic disc, which is formed around the third week of development. Now, if you don't forget, we want to look at how neural tubes are being formed from this trilaminar embryonic disc. Now, let's continue. This is the ectoderm, and this is the notochord. So at either side of the notochord, what do we have? We have the paraxial mesoderm, we have the intermediate mesoderm, and we have the two lateral plate mesoderm. At either side as well, we also have the paraxial mesoderm, we have the intermediate mesoderm, and we have the two lateral plate mesoderm, which is the somafluid lateral plate mesoderm, the one in contact with the ectoderm, and this one which is in contact with the endoderm is what we call the what? Splanchnic lateral plate mesoderm. Now, if you don't forget, the development of central nervous system occur around the ectoderm. Do you get? And development of central nervous system occur as a result of inducing action by the notochord. It is the notochord that induces the process called neurulation. which is the development of brain and spinal cord. How did the notochord induce lumination, and how the neural tube is formed? That is what I want to explain. Now, this notochord is going to secrete a growth factors. We call them cytokines. There are different types of factors that are going to be secreted by the what? Notochord. So this notochord is going to secrete a growth factor around this ectoderm in the midline. You get this ectoderm in the midline that is above it. That is where the notochord will secrete the growth factor. So when this notochord secretes the growth factor, you get, so these are the growth factor. It secretes it around the ectoderm that overlies it along the midline. And when it secretes the growth factor, what happens to the ectoderm? The ectoderm differentiates. Now look at it here. We have this to still be the what? The ectoderm. You get? Now at the middle part at which it secretes what we call the growth factor. Let's say this is where it secretes the growth factor. You get now? And this is the remaining part here. So we also have this to still be our notochord. This is the parietal lateral plate mesoderm, parietal mesoderm, the intermediate mesoderm, and the lateral plate mesoderm. Likewise, this same place. And this is our what? Endoderm. Now, if you look at it, as a result of the fact that the notochord secretes a growth factor around the ectoderm above it, what happens to the ectoderm? The ectoderm differentiates into another structure. Now, this part of the ectoderm now that differentiates as a result of inducing action of notochord is what we call the neural ectoderm. This is what we call the what? Neural ectoderm. Yeah. When we say neural ectoderm, what did I mean? It means that the ectoderm that will later give rise to our central nervous system. Then this part of the ectoderm that does not undergo differentiation is what we call the what? Surface ectoderm. Yeah, this is what we call the surface ectoderm, which is going to give rise to the skin and so on and so forth. Yeah. So if you don't forget, we now have the formation of neural ectoderm, the ectoderm that will give rise to our central nervous system. Now, what happens to this neural ectoderm? Because we are only laying emphasis on this neural ectoderm because it is the one that will give rise to our brain and spinal cord. So this neural ectoderm is going to become thickened. It's going to become flattened and appear as a slipper shape in order to become what we call the what? Neural plate. What do I call it? Neural plate. You get? So it is a result of the thickened and flattened action of the neural ectoderm. That is why the neural ectoderm becomes the what? Neural plate. It becomes flattened, thickened, and appear as a slipper shape. You get? That is why it's called the neural plate. And neural plate is formed around day 17. Yeah, around day what? Day 17. That is when we have the formation of neural plate. Now let's continue. Now what happens is that there's a junction at which the neural plate meets with the surface ectoderm. You get? You know, the neural plate is also the neural ectoderm, but because it has become differentiated into another structure, that is why we call it the neural plate. So there's a point here, this is the point, yes, at which the neural plate meets with the surface ectoderm. So what happens to that point? I just want you to take note of that point. You see this mesoderm at either side of the notochord, they begin to proliferate, they divide in number, and they push this end at which the neural plate meets with the surface ectoderm. They push the end up so that this lateral edge will elevate. I get what I'm saying? And this is how it looks like. This is our surface ectoderm. You get? And when it pushes the end up, okay, let me use it like this. This is our surface ectoderm. This, it becomes this. Also have this, and we have our notochord, the parietal, the intermediate, and the two lateral plate mesoderm. The parietal, the intermediate, and the two lateral plate mesoderm, and the what? Endoderm. Now, as you can see, that as a result of proliferation of the mesoderm pushed this lateral edge where the neural plate meets with the surface ectoderm. They pushes it up so that this part here can elevate. so that the lateral edge can what? Elevate. So that it can bulge into the what? Amniotic cavity. You know, above this ectoderm is what we call the amniotic cavity, and below the endoderm is what we call the what? Yolk sac. So this point here that is bulging is entering into the amniotic cavity. In order for this, as this point bulge up and elevate, what happens to the neural plate? The neural plate begin to invaginate. Can you see? Can you see the neural plate? It invaginates. And when the neural plate invaginates as a result of elevated action of the what? Surface ectoderm. What happens? The neural plate becomes a U-shaped structure. And this is what we call the what? The neural groove. This is what we call the neural groove. Yeah. The neural groove is also formed around day 18. So it is a U-shaped structure. That is why we call it the neural groove. Now, this point at which the mesoderm pushes up to elevate, this lateral edge at which they pushes up in order to form this neural groove is what we call the what? Neural fold. Neural fold. Yes, the neural fold is also forming alongside with the neural groove, which is at the what? 18. This neural fold, there's something I want to tell you about the neural fold. The neural fold is made up of two cells. It is made up of two cells. What are the two cells? It is made up of neural crest cells and the neural epithelial cells. Yes. So these are the two cells that are lying within the neural fold. You know, the neural fold is that lateral edge that elevates into the amniotic cavity, causing the neural plate to invaginate to form a U-shaped structure, which is known as the neural groove. So what happens? Don't forget that I'm explaining the development of neural tube, how the neural tube is being formed. Now let us go on. What happens next is that these two surface ectoderm want to approach one another. They want to come together in the midline. Do you know why? So that they can close up the neural groove. That is the essence of them coming together in the midline to bridge up this space. So as the neural fold, as the neural fold are approaching one another, what happens with the surface ectoderm? The surface ectoderm also meets in the midline. You know, there is a theory that says like cells fuse together. So as the neural fold is coming closer, the surface ectodermal cell at this place and the surface ectodermal cells at this place come together. And as the surface ectodermal cells come together, what happens to the neural groove? They also come together and close this up. This is the diagram here. So this is the surface ectoderm that have come together. They bridge up this space. And when they bridge up the space, what happens to the neural groove? The neural groove is like this. You get, you know. So as the surface ectodermal cell bridge up the space, what happens to the neural groove? It becomes deeper and begins to close this off to form what we call neural tube. But this neural tube here, it is an incomplete neural tube. You will know why I said it is incomplete. Then we also have this to still be our paraxial mesoderm. Okay, this is our notochord, paraxial mesoderm, intermediate, and the lateral plate. The paraxial mesoderm, intermediate, and the two lateral plates. And we also have our what? Endoderm. Yeah, so can you see now that when the surface ectodermal cell come together, the neural groove have closed this up, but it formed an incomplete neural tube. Then don't forget, I said there are some cells that are present within this neural fold. What are those cells? The neural crest cells and the neural epithelial cells. So as these neural folds are coming together to close up this neural groove, there are some cells that differentiate away from the neural fold so that they will not locate themselves in the surface ectoderm. Because if they did not differentiate away from the neural fold, what happens? They will be located in the surface ectodermal cell. What are those cells that differentiate away from the neural fold when the surface ectodermal cells are closing up? Those cells are what we call the neural crest cells. Yeah. So these are the cells here that they are located here. Let me make it very clear. This is the surface ectodermal cell. So these are the cells. These are the cells. So these cells at first, they appear as a group of cells. But later on, they divide into two to be located at either side of the what? Neural tube. So these are the cells there. They divide and locate at either side of the neural tube. And what is the name of those cells? Neural crest cells. These are the two neural crest cells. You get? They locate themselves bilaterally at either side of the neural tube. Now, if you don't forget, there are still some cells in the neural fold. You get? Those cells are what we call neural epithelial cells. Those neural epithelial cells will remain in the surface ectoderm when they bridge up the space. While some neural epithelial cells will also differentiate and enter into the neural tube. So don't forget that within this neural tube, there's also some cells. We call it neural epithelial cell. While there are some cells in the surface ectoderm also, we call it neural epithelial cells. Those neural epithelial cells that did not leave the surface ectoderm, that still remain in the surface ectoderm, is what we call what? Ectodermal placode. Yeah, it has a name. We call it what? Ectodermal placode. You see, these are the cells, undifferentiated cells of the neuroepithelial cell that are still in the surface ectoderm. So this ectodermal placode also gives rise to structures of the human body. It is the one that gives rise to the eyelids. Yes, it gives rise to the eyelids. It gives rise to the inner ear. Yes, and it gives rise to the sensory ganglia of cranial nerves. It gives rise to what? Sensory ganglia of cranial nerves. Sensory ganglia of cranial nerves. Yes, so these are the major structures that are forming from the ectodermal placode. You get? I just want you to know about that. There are those neuroepithelial cells that are still remained in the what? Surface ectoderm. While there are some neuroepithelial cells that are now located in the neural tube. You know, we need neuroepithelial cells in the neural tube so that they can differentiate and cause the neural tube to give rise to critical structures of the brain and the spinal cord. Now, I told you something about the neural tube. I said the neural tube is an incomplete neural tube. How can we know that the neural tube is incomplete? We are going to know via a section. We call it sagittal section. You know, when you undergo a median section, dividing it into two median half, that is when you know that this is not a complete neural tube. Now look at this. We have this. It is the continuation. So we have this to be the surface ectoderm. Okay. We have this to be the what? Surface ectoderm. Alright. Now, if you undergo a sagittal section, if you undergo a sagittal section of this, you notice that there are some parts of the neural tube that are still attached to the surface ectoderm. Do you get it now? And this is how it looks like. This is the neural tube. This is the neural tube. Now, if you undergo a section of this, you will see it to be like this. So this is what we call the surface ectoderm. Yes, the surface ectoderm. And this is what we call the what? The neural tube. You know, I said this neural tube is an incomplete neural tube. So how can we know that it is incomplete? It is when we undergo a sagittal section. So sagittal sectioning of this, let us know that there are still some parts of the neural tube that are still communicating with the surface ectoderm. So when these surface ectoderm are closing up and the neural groove is fusing together to form the neural tube only closes up around the cervical region in the fourth somal stage. That is where it closes up. That is where it fuses up around the cervical region. So this is the only point at which the neural tube closes up. That is why it is an incomplete neural tube. I think what I'm trying to explain here, you understand it very well. Now, let me make use of this paper as an example. Now, look at this paper. This is what we call the Let's say this is the ectoderm. Do you get it now? So when the notochord secretes a growth factor around the ectoderm above it, do you get it now? What happens to the ectoderm? It becomes the neural ectoderm. From the neural ectoderm, it becomes the what? Neural plate. So what happens to the neural plate? The mesoderm at either side of the notochord begin to proliferate and push the end up. This is the end at which the neural plate meets with the surface ectoderm. So this is the neural plate here in the middle. This is the neural plate in the middle. So at either side of the neural plate is what we call the what? Surface ectoderm. So what happens is that when these two mesoderm pushes up this place, then it becomes like this. Do you get it now? So when the mesoderm pushes up the point at which the neural plate meets with the surface ectoderm, this is how it looks like. Can you see the median part invaginating? Can you see the neural plate invaginating? So what happens is that the neural fold, this point here, this point is what we call the neural fold. So the neural fold wants to close up. Do you get it now? They want to close up, and as the neural fold is approaching one another to close up, what happens to the what? To the neural groove. It is also what? Closing up. And as the neural groove is closing up, the surface ectoderm is also approaching each other. So when the surface ectoderm approaches each other, they only fuse in the midline. Do you get it now? So the fusion only occurs in the midline. I get what I'm saying? That is in the cervical region. That is why I said this neural tube that is formed, this is the neural tube. Do you get? This is the neural tube. So the neural tube that is formed is incomplete because it only fused at the cervical region, around the fourth somal stage. That is what I'm trying to explain here. So if you look at the neural tube, you get, there are still some open ends of the neural tube. Can you look at it? There are still some open end of the neural tube at the cranial part and at the caudal part. This open end of the neural tube, it has a purpose. The purpose is that, I said above the ectoderm, there's something called amniotic cavity. So the fluid in the amniotic cavity can enter through this open end of the neural tube in order to nourish the neural tube until blood vessels has been established. This opening of the neural tube at either end is what we call we call this part here to what we call the what? Cranial neuropore. What do I call it? Cranial neuropore. Some people call it anterior neuropore. You can also call it anterior neuropore. Why this is what we call the caudal neuropore. Or you call it the posterior neuropore. Is it that you call this anterior or cranial, or you call this posterior or caudal? That is the reason for the neuropore, so that fluid from the amniotic cavity can nourish the neural tube. Then, as time goes on, there's something we call folate. Do you get? In a gravida, pregnant woman, when all this embryo is developing, they need folate in their body so that the folate can signal for the closure of the anterior and posterior neuropore, or the cranial and caudal neuropore. Do you get? Normally, the cranial neuropore should close around day 25. That is around 18 to 20 somite stage. Yeah, around 18 to 20 somite stage. While the caudal neuropore, also called the posterior neuropore, should close up around day 27. Yeah, 2 days later, day 27. That is around the 25 somite stage. Yeah, the 25th somite stage. Why this one is 18 to 20 somite stage. Yeah. So the neuropore needs to close up in order to form the entire complete neural tube. So when we have the closure of this neural tube, then at this stage, that is when we know that we have formed our complete neural tube. So this is our complete neural tube. Now, if you want to undergo a lengthwise longitudinal section of this neural tube, this is how the neural tube looks like. Yeah, it is something like this. Yes, this is the neural tube. This is the cranial part of the neural tube, and this is the caudal part of the neural tube. This cranial part of the neural tube will give rise to the brain. While this caudal part of the neural tube will give rise to the what? Spinal cord. At either side of the neural tube, what is present there? Wow. Neural crest cells. Look at it. Neural crest cells, they are located at either side of the neural tube. When we look at it in a lengthwise manner, these are the crest cells. These are the neural crest cells that are located at either side of the neural tube. If you don't forget, I told that there are some cells that are present within the neural tube. What are the name of those cells? They are the neuroepithelial cells. Yeah, they are present within the neural tube. So this is a complete neural tube. And when is the neural tube formed? It is formed around day 21. This incomplete neural tube is formed around day 21, but the complete neural tube is formed around day 25 when the cranial neural port is closed, and day 27 when the caudal neural port closes up. So this is the entire complete neural tube. Now there's something I want to tell you about these neural crest cells. Okay, before I talk about the neural crest cells, there's something you need to know, that if this cranial and caudal neural port did not close up on its normal closure period, you get, it can lead to neural tube defect. You get, when this neural port closes up early, or maybe after when it should have closes up, it can lead to birth defect. You get, so let us start with the cranial neural port. What if the cranial neural port doesn't close at day 25? It can lead to what we call cranial bifida. It can lead to what we call what? Cranial bifida. Yeah, cranial bifida. What do we understand by cranial bifida? Cranial bifida is a condition whereby the brain and the skull, it is kind of like, it is not fully formed. You get, that's what cranial bifida is all about. When we have the failure formation of the what? Brain and the skull, that's what we call the cranial bifida. Bifid, when something is not properly formed, that's what we call bifida. So that is cranial bifida. And cranial bifida can also be divided into what we call the meningocele, meningoencephalocele, and the last one, which is the meningohydroencephalocele. Yeah, so failure of the brain and skull to develop properly is what we call the cranial bifida, and it is divided into three. Meningocele. What is meningocele? From the word meninges, meningocele. Meningocele is the herniation of the meninges through a bony defect of the skull. You get, so you know, when there's a defect associated with the skull, most especially the occipital bone, you notice that the meninges will protrude out through those defect openings, and it will be located outside the skull where it forms a sac. We call it meningeal sac. That's what we call meningocele. This is what we call meningoencephalocele. Meningoencephalocele is a condition whereby when the meninges herniate or protrude out to form a sac, the brain will also passes through the opening defect and locate itself within the sac. That's what we call meningoencephalocele. Meninges and the brain protruding, herniating. That's what we call meningoencephalocele. The third one is what we call meningohydroencephalocele. At which the meninges have herniated to form a sac, the brain will protrude, and the ventricular system of the brain will also protrude along. That's what we call meningohydroencephalocele. They are caused as a result of failure of the cranial neural port to close at day 25. It forms the cranial bifida. It leads to cranial bifida. It also leads to anencephaly, whereby we have underdeveloped brain and incomplete skull. That is what we call anencephaly. Yes, this is what the cranial neural port can lead to, birth defect. What do you understand by anencephaly? When there's an incomplete development of the brain and skull. And this anencephaly can be divided into two. We also have the meroanencephaly and holoanencephaly. Meroanencephaly is a condition, a congenital malformation where there is absence of cranial valves. The calvarium is absent and there is exposed dorsal neural tissue of the brain. That's what we call meroanencephaly. But holoanencephaly is a condition whereby the entire skull is missing and the brain will be exposed towards the surface. That's what we call holoanencephaly. These are what can be caused. These are the birth defects that can be caused as a result of the failure of this cranial neural port to close at day 25. Yes, that is it. What if the caudal neural port doesn't close at day 27? It can also give rise to a birth defect called spinal bifida. What is spinal bifida? Yeah, we call it spinal bifida. That is for the caudal neural port. Spinal bifida is the failure of the spinal cord to be fully formed. You get now, when there is a defect associated with the spinal cord, that's what we call the spinal bifida. And the spinal bifida is also divided into two. We have the spinal bifida occulta and spinal bifida cystica. Occulta and cystica. Occulta, it is a mildest form of the spinal bifida where some mini structures within the spinal cord protrude through a defect opening in the spine. You get? Before you can know that somebody has spinal bifida occulta, there will be a hairy patchy area around the inferior region of the spinal cord. That denotes spinal bifida occulta. You get? It is not a severe form of spinal bifida. But you see, spinal bifida cystica is a severe form. Do you get now? It is a very bad type of spinal bifida. It is also divided into two. We have the meningocele and the myelomeningocele. So what do we understand by meningocele? Meninges. Annihilation of meninges through the defect opening in the spine to form a sac outside the spinal cord. It is just like the meningocele of the skull. While the myelomeningocele is a condition, a congenital anomaly, where the meninges annihilate through a defect opening in the spine, and what happens again? The myelin sheath, the nerves, also annihilate through the defect opening in the spine and locate themselves within the what? Meningeal sac. That's what we call myelomeningocele. Okay? So we also have a particular disorder, we call it rachis-chizis. Rachis-chizis. Rachis-chizis is a congenital malformation whereby there is a large cleft in the spine. There is a large cleft in the spine. And you know, there's something that surrounds the spine. We call it vertebral column. So those vertebral column that are surrounding the spine, it is not fully formed. They are confused, leaving the open spine to be exposed towards the surface. That's what we call the rachis-chizis. We also have myeloschizis and so on. So these are all the congenital anomaly, birth defects that occur if the cranial and caudal neural port doesn't close around its normal closure period. Lastly, there's something I want to explain about the neural crest cells that are located at either side of the neural tube. Neural crest cells are the ones that will give rise to the major peripheral nervous system because they are outside the central nervous system. This neural tube is the one that will give rise to our CNS, which is the brain, and the what? Spinal cord. So what are the neural crest cells we give rise to? The neural crest cell begins to migrate to give rise to critical structures of the embryo. When the neural crest cell migrates to the head region, especially around the pharyngeal arches, it gives rise to many structures. We call it the cranial neural crest cells. We call the cranial neural crest cell. Cranial neural crest cell. The cranial neural crest cell will give rise to what we call the bones of the middle ear. It will give rise to the what? Bones of the jaw. It will give rise to the thymus. It will give rise to the what? Odontoblast. Okay, can you see the structures that the neural crest cell are giving rise to? Where the neural crest cells, you see these cells, they migrate to different regions. When the neural crest cell migrate to the heart, we call it the cardiac neural crest cell. It gives rise to the what? Aorticopulmonary septum, APS, aorticopulmonary septum. It gives rise to the conotruncal cushion, CTC, conotruncal cushion, conotruncal cushion. It also gives rise to the valves and arteries of the heart. So when the neural crest also migrate to some certain region, most especially around the trunk, it also gives rise to structure. Do you get? It is the one that forms the dorsal root ganglion. The neural crest cells, dorsal root ganglion. The dorsal root ganglion is also formed by the neural crest cell. The sympathetic ganglion, adrenal medulla, melanocyte, they all come from the neural crest cells when they migrate to the trunk. There are even some neural crest cells that migrate around the vagal and the sacral cells. Do you get now? Around this inferior part of the body, and they give rise to what we call some nervous system, enteric nervous system. You get? It also gives rise to the postganglionic parasympathetic nervous system. All this come from the neural crest cells. That is why they are cells outside the central nervous system. Can you see? They are located outside the what? Neural tube. Because the neural tube will give rise to our central nervous system, which is the brain at the cranial part and the spinal cord at the what? Inferior part. Now, we have come to the end of this video, which is the development of central nervous system 1, the neural tube. Don't forget that at the end of this video, there's always a practice questions that will enhance and secure rigid knowledge on what neural tube is